Okay. A very warm welcome to the last day session of this workshop. And I hope the very good afternoon to sir also. And I hope sir last 3 days workshop will be a very fruitful. Actually, it is you provide really a skillful knowledge to us, and it will really a first time experiencing. We are really at hands-on training on such a software, freely available software, that we really don't know how to work on it. Although we know, but we don't know how to work on it. So we are really looking forward to our 4th day session, which is the last day session, and it's very important as a in the aspect that you will be really teaching us the molecular docking, and which is a very important part of the research. So, sir, I request you to continue the session on the for the molecular modeling. Thank you, sir.
Thank you very much. Thanks for the introduction. You can see my screen?
Yes, sir. It's visible.
It's visible, sir.
Yeah. So we have been talking past 3 days. The entire left-hand side, the ligand-based design, pharmacophore-based approach, and QSR-based approach, and various drug likeness property, ADME, bioavailability, GA absorption, various rules like Lipinski's rule, all those. No? So I hope you are thorough on that, on the side, the left-hand side of it. So we do use the ligand-based design if we do not have much knowledge about the target. Because if you want to know the target, it's not very simple. In real life situation, suppose we isolate, for example, a tumor, and then we run a 2D gel electrophoresis. There will be 40 or 50 proteins mixed together, you know. So my protein of interest, if I'm going to isolate using some chromatography, I should have enough amount to get sequence. And then if I want to get a 3D structure, I should have enough amount to get a crystal. So it's not a joke, actually. It's very difficult, especially for human related targets from the clinical samples. Animal samples are much easier because it's possible to generate various animal models. Suppose you want to generate a tumor based animal model, it's easier. So you can be able to collect enough sample, but even that is not so easy because the separation of proteins, getting the pure protein, sufficient quantities, and then running the X-ray crystallography and then trying to get the structures is a very time-consuming process actually, and difficult process. It's almost like it's a PhD actually. That's why there are not much of human targets available. You may have animal targets for most of the diseases, not all, most of the diseases. Still, there are 80% of targets are not available for diseases. Please remember, almost 80%. And for human, still much less. So, target-based design, we use some protein analogous to that. So, it's not really 100% exact, actually. We use something analogous. So that's why we carry out something called homology modeling. Analogous structure we use, and then we build the protein of our interest. Okay, that's called a modeled protein. So the modeled protein will not be the real protein. So that is one of the biggest assumption when we do docking. So don't think that when I do docking, I get correct answer, because like I said, 80% or 85% of proteins targets involved in human diseases are still not crystallized. You may have it for some other mammal actually. So we create the homology of the protein of our interest, and use that as our modeled protein, and then we do docking of small molecules. Okay, that's the main, that's what is called homology modeling. And then you do the docking of uh, various small molecules, desired molecules, and then you may select a few which looks very promising with respect to the binding. And then of course we have to do the filtering. Don't forget this. This is always going to be the most important. Whether you do this path or this path, this is important actually. We can combine both the paths also. Suppose I run a pharmacophore like we did on the second day, pharmacophore modeling, we got about say 1 lakh or 2 lakh compounds, right? And then with that 2 lakhs, you did some Swiss ADME, maybe you got 10,000 or 15,000 compounds. Suppose you know the target, then you can dock all the 15,000 molecules and see which ones dock well, and then select those. So that way you can combine pharmacophore modeling, that is the left hand side with the right hand side also, if you know the target. If you don't know, of course you can't do that, but generally that's how we do. We combine the left hand side with the right hand side. So we do a pharmacophore, we identify lakhs of molecules from zinc, and then maybe do the filtering. Maybe you get 10,000 or 20,000 molecules, then dock all of those 10,000, 20,000 to the target of interest, okay, and then see which ones dock very well with respect to some known control, which one docks not so good. So select those. Maybe you will end up with thousands of molecules. So you combine both the ligand based and target based design. Okay. Basically, if you look at the structure of a protein, protein has a primary structure that's a linear sequence of amino acids. Okay, AG, like that, amino acid sequence. This is easy to get. The amino acid sequence of a protein is easy to get by running LC-MS-MS. So we purify the protein using, say, 2D gel electrophoresis, and then we run a LC-MS-MS. No, we have to digest it. That means you have to break, because that protein will be very, very long. So we break it into pieces. Okay, digestion it's called, and then take each one of these and then get the sequence using LC-MS-MS, and then assemble the whole thing. So sequences are easy to get, much easier. Easy means it's not so easy, but it's easy. But getting a 3D structure is difficult. So the primary structure of a protein is the amino acid sequence. Then you have secondary structure. This is alpha helices. That means it will be looking like a round and round, going like a helix, right? Helix. Beta sheets and turns. They're all connected by hydrogen bonds between the carbonyl O and the amino H. Okay, these are called the secondary structure of a protein. Okay, then the tertiary structure is a three-dimensional of the protein actually. Okay, that is this tertiary structure. And finally, you have quaternary structure also, where you can have many units are assembled together. So many cases, these proteins are polymers. That means they will have 2 subunits, the same thing, 2 units or 3 units or 4 units, like that, assembled together. Okay, so that's the quaternary structure. Okay, so the proteins are basically has 4 different classes of structure: primary, secondary, and tertiary, and quaternary. Okay, so if you go to PDB database, generally you may get the polymeric. That means many subunits may be connected. So if you want to do some docking, we remove those all the subunits and take only one subunit, so that docking becomes easier, and then we do the docking. Okay, that's how the whole protein sequences are aligned. So if you have a primary sequence of amino acids, no, A, G, C, T, A, G, whatever sequence, we can compare this primary sequence with the primary sequence of another protein, and we can see what is the sequence similarity. Okay, we'll look at that also later. Okay, this is the structure of a protein. So if I have a 3-dimensional structure of the protein, then what do you do? Life is easier. So we try to dock a lot of small molecules into the active site of the protein, and then we get the binding energy. So it's very simple if I have the structure. Okay, it's like putting, finding out the best key for a lock. So this is the lock. So I put in lots of keys. Some of them bind very well, giving you some binding energy. Some of them bind badly, giving some other binding energy. Okay, so this is very simple. Suppose we do not have a structure of the protein. Like I said, most of the protein structures are not 3D structures are not available. We will have only 2D sequence, primary sequence. So we build the 3D sequence using some template. Okay, so that is like making the log, building the log. The homology modeling is like building the log. And then once you have the log, you can, like the previous page, put in lot of molecules inside and see which molecules bind well, which molecules don't bind well. So if you don't have the log itself, that means if you don't have the 3D structure of a protein, then we build the 3D structure, like making your log. Okay, that's called the homology modeling. And then you fit in different ligands. Now, how do you build this log, or how do you get the 3D structure? So what I will do, I will compare the 2D sequence of my protein with the 2D, with the linear amino acid sequence of my protein with other amino acid sequences available, and then see which one is closest. And then what do? I will take the 3D structure of that and then build my protein on. Okay, so I will take that as the template and I will build the protein, which is called modeled protein. That's how we do. That's where homology modeling comes in. There is another approach. Oh, I don't have that approach. There is another approach also. If we know many keys open, we can try to sort of visualize how the lock will be. Same thing. So if I know many ligands dock very well with the protein, we can also build the structure of the active site of the protein. That is also another approach actually. Okay, just like we do homology modeling with the ligands alone, we can also think of doing homology modeling for the 3D structure of the active site also. And so these are various approaches by which target-based design is done. Here, the target is your protein or enzyme or whatever it is. Okay. Now, like I said, there are about 50,000 non-redundant protein in the protein data bank. Yesterday I showed you the protein data bank, PDB. Okay. Now, if you look at the protein sequence, that is the amino acid sequence that is present in UniProt or SwissProt, it's called UniProt, SwissProt, there are 5.5 million. Whereas 3D structures, only 50,000 is there. Okay. Because like I said, it's difficult to get the pure protein, crystallize it, run XRD, and then get the 3D structure. So lots of protein sequences are there, amino acid sequences there, but you don't have the 3D structure. Okay, it's almost like 1%. That's why we do homology modeling. Many cases you'll do homology modeling. You may have the target for certain type of cancer, bone cancer, in animal 3D structure of the protein in rat. You might not have the corresponding one for the human, but you will have only the sequence, amino acid sequence. Like that, you may have certain viral targets in animals. You might not have it in human. Okay, that is why we have very little, as you can see here. And even this 5.5 million is very small when compared to the number of proteins in the earth actually, because there are so many organisms, 5 million organisms, and each organism may have about 5,000 genes. So if you multiply that, there are so many proteins which we don't know. Whatever proteins that is present in SwissProt is only 5.5 million, which is very small when compared to 5 into 50, that is 25,000 million. 25,000 million proteins, possible proteins. So we still don't know so many proteins that is present in our earth. Okay, now so protein sequence, I have to get the 3D structure. So I do this homology modeling actually. That is how we do. Actually. Protein sequences are there in UniProt or SwissProt database. If you remember yesterday, I talked about this particular database, SwissProt or UniProt, for example. So each protein sequence has a name. Similarly, in 3D protein 3D database, that is protein data bank, again it has a name. So this also has a name. So the number P09917. This is a human lipoxygenase 5. This converts arachidonic acid. This is in the leukotriene pathway of the inflammation. This leads to asthma and so on actually. Okay, so this is called a lipoxygenase enzyme, 5 lipoxygenase. This is the sequence for the human, which is stored in the database called UniPro. And also it's called SwissPro. So a lot of the information is done given. Okay, so if you come up with a new protein from some system and you purify it and get the sequence, amino acid sequence, you may be able to put it inside this database. Okay, let's go for it. So, so many sequences are there, 5.5 million, and only 50,000 available in protein data bank. Okay, that's a big shortage. That's why we do homology modeling. And like I said, if you want to get a 3D structure of a protein for some human disease, if you are going to work on it, that's a PhD project. Okay, so we do homology modeling. Okay, what something very interesting you need to know. The structure is more stable and changes much slower than the sequence. So if the protein sequence changes quite a lot also, the structure is maintained. That's the beauty of the concept of homology modeling. Okay. So the amino acid sequences can change a lot, but the basic structure of the three-dimensional structure is very slow in change. Okay, this was identified by this group, Chatia and Lesk, 1986. Okay, and later again quantified by another group. That is why even if the sequence identity is low, 40%, 50% with respect to sequence of protein A and protein B, still the 3D structures is conserved. Okay, that is why we can do homology modeling. Otherwise, we cannot do. So even if there is a sequence comparison of about 40% only, we can still assume protein A will take the structure of protein B. That is how the evolution has happened actually. Interesting. So they still fold into similar structures. So it's not that sequence identity has to be 100%. Even 40%, such low number sequence identity, still it will form a 3D structure of the same actually. Okay, this is a very important graph actually, picture. So if you are aligning two amino acids, suppose I align 150 say amino acids, and I find a sequence. I compare the amino acid of say 150 residues with another set of sequence, and even if I find only 40% similarity, then I can use the 3D structure of that protein as a homology model, as a target protein, as a homology model to build this. So as low as 40, even 33% can be considered as safe actually. Okay. So that's a very interesting advantage which has happened in evolution. That is why we can do a homology modeling. Okay, so if I say, for example, I am comparing 150 amino acids, the 150 amino acids of another protein whose 3D structure is known, and I get about 35% similarity, identical. Then I can say protein A will have a 3D structure similar to protein B. So I can use protein B as a template to build my protein A. Okay. This is the concept, entire concept of homology modeling. Okay. So there is a program called BLAST, which will compare sequences, okay, and then say what is the identity. We will do that also. Don't worry. So if we are comparing a sequence of protein A and sequence of protein B, okay. So if we are comparing, say, 300 amino acids and 150 amino acids match of sequence A with sequence B, then we can say 50% identical residues. Okay. And sequence A is called the target, sequence B is called the template. Whichever 3D structure is aligned, we'll call the template actually. So the BLAST takes care of all this. It will bring together, it will do all those, and then give us some number. And then we will select the one which has very high identity with respect to the amino acid comparison. We will take that as our template and build our protein of our interest. Okay, that's how we'll do. So just to give an example, sequence A has 320 amino acids. Sequence B has 450 amino acids. Okay. Now when we do BLAST, that's a software which compares these two sequences, 320 and 450, and then 100 amino acids are identical. Then the percentage identity is 100 by, you have to take the smallest one. So 31.25%, they are identical. Okay, in that, if some of the amino acids are similar, as you know, total amino acids, many of the amino acids, hydrophobic, hydrophilic, all those. So there are similarities between amino acids. Okay, if 23 amino acids are similar in chemical properties, then with this 100 we can add. Okay, then that comes to 38.44, which is called similarity. So identity is this, similarity is this. So identity will always be less than similarity. Similar means it's not exact, whereas identity means exact actually. Do you understand? So if some amino acids can be similar because they may have similar properties, hydrophobic nature, hydrophilic nature. And so when you do a BLAST, it will give you a sequence identity. And then, as I said, where you have a highest identity, we will say those two sequences are matching. And then we can do the homology modeling. Okay? So this is the definition of sequence identity. This is the definition of sequence similarity. Okay, so how do we do this? I have the sequence. I don't know the 3D structure. It's like I did some research and I isolated a protein from some clinical. I don't have the 3D structure. So I take a template and compare. It's called multiple sequence alignment. There are many softwares, other BLAST and all that. Okay. This Swiss model will do. So what we do is we will compare the sequence, and like I showed you, about 30–40% sequence identity good enough. Then we build the model for my target based on the template. That's called initial model building. And then I have to do some model refinement and then model validation. We will look at all these things. Okay, then what is the active site? Maybe we can look at literature and get the active site. And then you take all the ligands and dock, and get some binding energy or docking scores, right? So you select those which have good binding energy. This is the entire concept of target-based design. So you will have the sequence alignment, and then identifying the best ones, sequences, and then we can build the protein. That's called the model protein. Then refine it. get it ready, and then take all the ligands, dock it. There are many softwares out there. Autodock is free. I'm going to show you another software, SwissDock. These are Gold, Glide, MOE. They're all chargeable things. Okay. Sequence alignment, there are no paid software. Everything is free. Blast is a free software. And then then we can do a molecular dynamics also, just to see how this docked ligand is stable. Does it come out, or is it very stable because of the hydrogen bonds and so on? So we can find the stability by doing a molecular dynamics of the target, the ligand protein complex. Okay, so we are going to look at all these. Okay, what are the drug targets? It could be enzymes, receptors, ion channels, transporters, DNA intercalating agents. So many. Okay, this could be your target. It's not only proteins, enzymes, they're all called different. And then your ligands could be inhibitor, reversible inhibitor, irreversible inhibitor. Receptor means antagonist, agonist, and channel means blockers, transporters means uptake inhibitors, DNA means intercalating agent, minor grooves, binder, antisense drugs, like that actually. So this left-hand side is the target, the right-hand side is your ligand. But they have different names depending upon what the left-hand side is all about actually. So we can have reversible, irreversible inhibitor. So if you take enzyme, if you have done a course on enzyme technology, or if you have studied about enzyme, you can have competitive inhibition, non-competitive inhibition. So you have the enzyme, there is a substrate, goes and binding to the active site. So forming the enzyme substrate complex, forming the product. This is how enzymes work. Okay, for example, if you take a lipoxygenase enzyme, arachidonic acid is the substrate. It is giving it 5-HETE, which leads to asthma. Or if you have a cyclooxygenase prostaglandin synthase, arachidonic acid binds to it, and it gives out prostaglandin H2 as the product. Okay, that's the function. So when you design a drug, you are designing a drug which will go and bind to that active site. Okay, so the substrate will not go into that. That's called a competitive inhibition. So a drug, a competitive inhibition drug can go and bind. So what happens? Your substrate cannot go and bind. So it's blocked. Substrate will not go and bind, so the product is not formed. So there are drugs which are working as a competitive inhibitor, which will go and bind to the same place as your substrate. You can also have non-competitive inhibitors also, not to the active site, but these some drugs go and bind somewhere else, and the enzyme shape gets distorted. So once the enzyme shape gets distorted, substrate cannot go and bind. They are called non-competitive allosteric inhibition. Okay, so in competitor, enzyme goes and binds to the same site as your substrate, so the substrate cannot bind. In allosteric or non-competitive, enzyme goes and binds to some other place, so the shape of the enzyme is disturbed, so the substrate cannot bind. That's called non-competitive inhibition. Okay. Okay, so this is how drugs are designed. It could be a substrate inhibition, competitive inhibition based, and allosteric based. And yesterday we looked at the protein data bank, different types of protein structures, right? For example, 3V98. Put protein data bank 3V98 type 3V98. 3V98, what is that? 3V98 is a 5-lipoxygenase, which is involved in leukotriene pathway for asthma. So the structure is there. We can see what it is. Is a yeah, human 5-lipoxygenase, we have 15 lipoxygenase point mutation. Okay, so you will have many lipoxygenases, 5-lipoxygenases in the database, because many groups would have crystallized lipoxygenase with different ligands bound to it. Actually, if you see helical assembly, it's some other complex of Pseudomonas aeruginosa. That's a bacteria that's involved in lung infection. Look at another one. So you'll have many lipoxygenases, Pseudomonas aeruginosa, and then limited of the origin of proteasome. Like that, you will have many possible actions. Okay. 5KIR, and that is cyclooxygenase 2. Cyclooxygenase 2 and ions. This is Vioxx, is a commercial drug. You must have heard this in our Pharmacokinetics model also. QSA also, where this is a commercial drug, and this is bound to human COX-2 enzyme. So PDB database contains 3D structures of protein with some number as an identity number, right? Okay, so when the ligand goes and binds to a target, generally it doesn't form a covalent bond. There will be only non-bonded interaction, electrostatic forces, hydrogen bonds, van der Waals forces, dipole, ionic forces. These are called non-bonded interaction. That's how the ligand goes and binds actually. So hydrogen bond plays a very important role for the ligand to orient itself and fix nicely inside the active site. Okay, so this is how ligands goes and bind to a target. So they are called non-bonded interaction. Okay, so when the ligand goes and binds to a target, you have some binding constant, the minus delta G by RT, right? Delta G, as you know, is delta H and delta S. So delta H is the heat, and this is the entropy. Entropy depends upon the flexibility of the ligand. If it has got too many rotatable bonds, it will have too much flexibility actually. And then K will be large. If K is large, that means binding is not very good actually. If you look at covalent bonds, generally covalent bonds are very strong, as you can see in kilocalories. Whereas non-bonded energies, and they are be very small, 1/10. We will see those. So any typical covalent bond, look at the energies, 80 going right up to 165, and also the length of covalent bonds are much smaller when compared to say hydrogen bond or electrostatic forces. Okay, high bond energy, short distances. High directionality. That means, as you know, it's carbon sp3. The angle is very fixed, right? 108 degrees. Then water, if you see, 120 degrees. So directionality is very fixed. Whereas electrostatic interactions, directionality is not fixed. Hydrogen bond, a little bit directionality is very important, but other non-bonded like electrostatic van der Waals, they are not very fixed actually. Okay. Hydrophobic, less than 10 kilocalories. When compared to, if you see the covalent, all 81 to 160, hydrophobic is less than 10. Electrostatic, 1 to 20. Hydrogen bond, pi aromatic stacking, van der Waals. All these are non-bonded interactions between the ligand and the protein. They are low energies, poor directionality, except hydrogen bond, all of them. It's not that it has to maintain a particular angle, exact angle at all. And then you also have salt bridges. Okay, that is ion pair or salt bridges. That is, if you have say N plus on one side and O minus on the other side, that forms a salt bridge. So the distance is 5 angstroms, within 5 angstroms. So this is very long. When you look at the covalent bonds, very, very short actually, right? So salt bridge. But the angles are very, very less. I mean, sorry, the energies are very low actually. So if you have two ions, and then the energy, if the charge is Z1 and charge is Z2, this is the equation for the energy actually. Energy, the charge of 1, charge of 2 divided by epsilon r. Epsilon is the dielectric constant of the medium. Okay. So if you look at the water, dielectric constant is very high when compared to say CdCl3. Okay. So water dielectric constant will always affect electrostatic energy because it's very, very large, which comes in the denominator. So the electrostatic forces are going to be much less. Ion dipole interaction. So you have an ion here and you have a dipole here. Okay, plus and minus, that's a dipole. So there is an equation for the energy in ion dipole interaction. Dipole, dipole, two dipoles interacting. So there is an energy for that. This is weak. Ion dipole is weaker than ion-ion interaction. Long range, some directionality, can be attractive or repulsive. Okay, whereas if you look dipole, dipole, very weak energies, very long range interaction, some directionality. Van der Waal interaction. Okay, Van der Waal interaction, you have terms like R power 6, R power 12 in the denominator. Okay, bond energy is very weak, very short range. And if the distance is more, because it is in the denominator, the energy numbers go down very much actually. So, whereas van der Waal, there is always all atoms and molecules will have this van der Waal interaction, but they have to be very close to each other, and the energy is very weak actually, as you can see here, 0.5 kilocalories. And then you have the same thing, this portion and this portion, and that this is called a London equation. Okay, this is for the hard sphere repulsion between atoms. This is the weak attraction soft. So look at these energies for water, cyclohexane, protein, and so on actually. Hydrogen bonds. Hydrogen bonds is very, very important. You can see some energy values between various amino acids here. Right? And the structural stability of the ligand protein is mainly determined by the hydrogen bonds. Protein stability is, as you know, when you go to secondary structure from the primary sequence, the amino acids are held because of the hydrogen bond. So hydrogen bond is very, very important actually in a protein structure. So hydrophobic interaction makes major contribution for protein stability. Hydrogen bonds very important for side chains and peptide groups. Disulfide bonds. We have sulfur groups present inside. They contribute to protein stability. And so on. Charge interactions are there. Salt bridges are there. So all these make the protein interaction, protein stability, and protein ligand interaction. So hydrophobic interaction contribute to 60%, hydrogen bonds to 40% for protein stability. Okay, this is a very important point. Another problem of instability is rotation around many bonds in a protein. Okay, so too many very flexible ligands, very flexible proteins always have instability problems. The ligand does not bind strongly with the protein. So when you run a molecular dynamics, after some time you can see the ligand leaving the place actually. Ligand leaving the place. Okay, we are going to use something called a Swiss model, okay, which is going to do the homology modeling, because as I said, homology is the first step in. Okay, we will, for example, take this sequence and we will use the Swiss model to use the homology. Swiss model. It's very good in the sense we can first do your protein sequence comparison. We can select the best one which has a sequence. Then we can do the 3D structure of the modeled protein using the template. So all these can be done. Okay, so it's run on a web server. Okay, right? Okay, so imagine you have discovered a new protein from some human disease actually. So if you have the sequence, you can put in the sequence here. Can you see this page? Can you see this page?
Yes, sir, it's visible.
Visible. Okay, so imagine you have isolated a protein from some disease from your lab or from your hospital collaborator, and then you purified, and then you got the sequence of the protein using LCMS, MS. And if the structure, the 3D structure is not there, so you want to do a homology modeling and see which protein is closer in the sequence. You can put in the whole sequence here. Okay, but what I am doing is I am just putting some uniprot. I put in this particular one, which sequence of prostaglandin synthase 2. Okay, prostaglandin synthase 2 enzyme. It's also called the cyclooxygenase 2 enzyme involved in the inflammation and the prostaglandin pathway of inflammation. So I just put in there. Okay, so once you put in that, the sequence I put in there, so the sequence is automatically copied actually. Okay, sequence is automatically copied. But if you have discovered a new protein and got a sequence, you can put in that. Once I do that, I search for templates. That means I am looking at many protein sequences and seeing which one has the highest sequence comparison. Sequence identity. Search for templates. Okay. So it will take some time because it goes through lots of protein sequences and running. Please wait. It will take quite some time, and it will give out based on the sequence similarity, sequence identity, and similarity will also be given. There will be something called E value, so it will arrange it actually. It says if you want to come back later, yeah. So it will run for some time, and then it will give you the sequence. So this is the sequence, amino acid sequence of this particular enzyme called a prostaglandin synthase 2. Okay, you can see the amino acid sequence. So if you have come and discovered a new protein, got your amino acids, you will have this. So you can put it inside and then start looking for sequence similarity. Okay, I think it's done. So what it has done is, whatever I gave, it came up with large number of results. It gives you a sequence identity here. See 88.36. Just look at this column. 99.3. And then it gives you crystal structure. What is this? Crystal structure of aspirin bound to cyclooxygenase 2 enzyme. Okay. So we can sort it based on GMQE. So 99% identity. 99.5% identity, even 100% is there, because I gave a known protein sequence. It is already there. So if I have a new protein which I have discovered, you will not get 100%, 99%. You may get 80, 70, 60, whatever it is. If it's very, very novel disease and novel protein, you may get 40, 50, 60 like numbers actually. Because I have given a known protein, I'm getting a big number. Okay, so it keeps giving a large number of proteins. It's giving the sequence identity percentage, and it also gives you how that protein 3D structure of the protein is obtained. X-ray crystallography 2.1 angstrom. Okay, since accurate, and then you have X-ray with 1.7 angstrom. And then X-ray crystallography, how it was determined. You can go to that structure. There will be reference papers. You can see more of them also. Okay, so it gives you a lot of other information, right? Sequence similarity is 62%. Okay, we come up. So so this particular protein here, we have identity of 99%. Here you have this particular crystal structure. You have a palmitic acid bound to this enzyme, cyclooxygenase 2 enzyme. Okay. And then this one, if you look at it, again, palmitic acid bind to cyclooxygenase 2 enzyme, and naproxen bound to the enzyme, this particular protein. And then this is a murine cyclooxygenase. That means from mammal cyclooxygenase. Diclofenac. This is an anti-inflammatory drug bound to the cyclooxygenase 2 enzyme. This is murine again, bound to a cyclooxygenase 2 enzyme. Like that. Okay, this is one compound called SC754. It does not have a name, bound to this enzyme cyclooxygenase 2. This is a salicylate. Just like after all, aspirin is acetyl salicylic acid. Salicylate bound to human cyclooxygenase 2 enzyme. So you have a sequence similarity of 88%. Vioxx. Vioxx is that drug bound to the cyclooxygenase 2 enzyme. And it has got a sequence 88%. So I can take maybe this one because I want to work on human cyclooxygenase 2, and this is a commercial drug in the market. So you can select based on what you want to work. Okay, that's very, very important because there are so many cyclooxygenase 2. There are some human, there are mammals, and then there are some commercial drug. There are some salicylate, there are palmitic acid. I am more interested in commercial drug. So I will select this chap maybe, and then I will say, I can say build model. Okay, I can say build model. So what have I selected? I have selected a human cyclooxygenase 2 with the Vioxx bound to it. Okay, it's identified 15 templates. Different types of researchers worked on different ligands bound to the cyclooxygenase, human cyclooxygenase, mammal cyclooxygenase. So many are there. Depending upon what you want, you can download that actually. Now, I'll click on build model. So it will take this 3D structure of the protein. Where is that? 3D structure of the protein. This will be called a template, and then it will build my protein sequence that will be the modeled protein. Okay, this will be the template, and when I build my protein, that will become the modeled protein. It's not a real protein, right? Real protein is through X-ray or NMR or something, whereas you are modeling based on a template. So build model, I can click on this. It will build my protein based on my sequence, but using that template. Okay, it's called build model. So it's building. So it's done very fast. So we can now assess the built model. There are many tools. There is something called Ramachandran Plot, RMSD values, and so on actually, just to see whether my protein has been built properly. Okay, structure assessment. Okay, so it has built my protein. This is called Ramachandran Plot. Okay, have you heard of Ramachandran Plot? Anybody? Has anybody heard of Ramachandran Plot? Yeah, good. One person has heard of Ramachandran Plot. That's good. So this Ramachandran plot, when the protein is assuming a three-dimensional configuration, uh, it's got an angle of that angle is called phi. In the third dimension, that angle is called psi. So it can have only certain values of phi and psi acceptable, and you check your protein and see whether it is in the acceptable region. If in this particular case, most of the points, the phi psi angles are in the acceptable region. Only very few are not in the acceptable. But if you have more points outside, that means that protein will not be really realistic, because Professor G.N. Ramachandran, okay, he's Indian. He worked in first he worked in Madras University, then he worked in IISc Bangalore, and he developed this particular knowledge of acceptable region for angle phi and psi of a protein. So that this is used quite a lot everywhere in bioinformatics. In fact, he also got the 3D structure of some of this molecule, the helical structure, okay, alpha helix structure. He got it. He should have got the Nobel Prize, but because of some British politics, he did not get the Nobel Prize. Okay, he was the first person to get the phi psi acceptable regions in a protein. Those days there was no computer also, so Very great. Like I said, he did not get the Nobel Prize because of the politics of the British. So this is a very important uh, plot which tells you whether all the angles of your protein, which is modeled, the phi and psi angles of each one of these amino acid connections are falling within the acceptable region. So in this particular case, only one or few are not falling in this acceptable region. You can even find out where it is. Okay, so it's giving where it is not falling. So at this point, where it is not falling. So this is a, so you have to check whether your protein is in the acceptable region or not in the acceptable region. And so on. Okay, that's the validation of the protein. And then we look at many things, okay? Many other things. So clash code. If it does not, if one phi psi it's outside, that means that particular amino acids one above and below, they may be clashing with each other. They might not be. Ultimately, the amino acids should not be clashing with each other, very close. They should have that distance. That is what this phi psi tells you actually. So 96.6% of the angles are in the acceptable region. So that you need to check it out to see the modeled protein is okay. And then we look at the residues and the predicted regions also. This is called the local quality estimate. We should not have very large deviations here. The deviations are very small. So again, the protein is quite stable. And then we also look at the mean residue quality check. Okay, so in this portion, those two are not matching. Yeah, where the chains are not matching properly. So this is called a quality check. You just have to go through everything just to see. That's called structural assignment. Okay, so you now have modeled this protein. This is called the modeled protein using a template of human cyclooxygenase 2 bound with Vioxx drug, because that's what I selected for modeling actually. So you can download this structure, PDB, that is protein data bank, for your modeled protein, and you can use it for your future docking actually. So this is the approach for doing a homology model. You understand? So we start from Swiss prot or Swiss model, sorry, not Swiss prot, Swiss model, and then we give the amino acid sequence of the protein of your interest or protein which you have discovered, and then you see search for template. It will search and you can arrange it in the order of uh, sequence identity, then sequence similarity, for example, and then you can select the best template depending upon what is your research is going to be, and then we say build model. It will build the model. It will give you a protein structure. Okay, then you can check the structure to see whether all the Ramachandran plot or RMSD, everything looks good. You download that 3D structure. That will be the modeled protein, which you use it for your docking purposes. That's how the entire process is all about, actually. Okay, anybody has any doubts on this? We can even use two proteins as a template. And so when we do the so it came up with many, right? So sometimes if you want, if you feel, you can use more than one to build your model. I can mark two protein and then I say I want to model my protein using two templates. That's also possible, actually. So I'll get a modeled protein which will consist of both the proteins average, almost like an average of the two protein templates I have selected. So you can do that also. It's like if I want to build a new car by combining the design of say Maruti and Ford, so I end up with a new design, an average of both, actually. Okay, that's how the entire concept of looks like.
So one of the participant is asking about the docking, that what is the difference will be there if we dock without model PDP?
Where will you get your protein? Where will you get a protein? Tell me. Suppose I have been explaining all this so long. In many cases, you don't have human targets. You will have only animal targets. 3D structure of human protein, right? You will have animal protein. Either you can just dock it, but that's not exactly correct, right? Or if you have come up with a new, you have identified a new protein from your clinical research, and you got a sequence, the amino acid sequence. You don't have a you don't have a PDB structure, right? So what will you do? You have to do a homology modeling. Right? So two reasons you have to do homology modeling. One is in many cases I will not have the 3D structure, PDB structure for a human target. I may have it for from a rat or mouse or something. So either I use it or I try to model for the human based on the amino acid sequence. Number one. Number two, I have identified, I have isolated a new protein from my clinical studies, okay, for some disease with the collaboration with some medical center. Now I got the protein sequence using LCMS-MS. I have the sequence, I don't have the 3D structure. So I have to model that. Right? Two reasons we have to model. And in many cases, you will not have human targets. If you spend some time, you will see many cases you might not have the human targets because it's difficult to get enough protein from a diseased person, whereas it's easy to get it from animal because we can have a genetically modified animal. Then all the animal studies based on that, we can isolate that protein. If you want to, you suppose there is a tumor you want to study, I can develop such tumors on animals, then isolate the protein. I can't do that on humans, right? So that is the problem. So in many cases, you will have animal-based targets. You will not have human-based targets. So all of you try to spend some time on PDB database and see what you have, what type of protein structures are there, okay, what type of diseases, human targets are there, what type of diseases. It's very important for you to know. Okay, so this is how you do the homology modeling. So in most of the cases, we'll be doing homology modeling. Okay, most of the cases. I want to study a particular type of cancer on a human, blood cancer. I may have the target protein in animals. I might not have for human. Then you have to do homology. Or I may have for human, but it will not be very relevant because if so, lipoxygenase, formic acid bound to lipoxygenase, some drugs bound to some open form, so many things will be there. So something which is very relevant for me might not be there. Then you have to do the homology modeling. So this is going to be part of it. Before docking, many cases we will be doing homology modeling. Okay, now you have done the homology modeling, you got a protein, you download it, and then you can do docking. There are many commercial softwares are there, and there are many free softwares out there, very good softwares. Autodoc Lina is there, SwissDoc is also there. I'm going to show you SwissDoc for docking. Yeah. Let's look at it. Aspirin with the cyclooxygenase 2 enzyme. So I'm going to use cyclooxygenase 2 1PXX. What is this 1PXX? 1PXX is a this is a structure of a drug called diclofenac. It's used in inflammation bound to the cyclooxygenase 2. Okay, so that's that. So I'm going to use that 1PXX with aspirin. I want to see that. Okay, so SwissDoc. This is an online docking server. The results will come to your email ID. Okay, so this is SwissDoc. It's called Subit docking. Okay. Okay, sometimes, many times when you download from PDB, the protein will not be really good for docking. PXS, so will not be good enough for docking. Because PDB will not have hydrogens, PDB will not put in charges distribution. We need all those when you are doing binding energy calculation. PDB may have some solvents present. So we have to do something called dock preparation. Dock preparation. So we have to do some dock preparation. So I will download this PDB format. Okay. Then I have to take it to the UCSF camera, because most of the docking programs can accept only 1,500 sorry, 15,000 atoms. And like I said, they will not be having hydrogens. They will not be having, and also, see, COX2 is a dimer, so all those problems will be there. So I need to prepare the docking. For example, if you see, it's got so many chains, COX2, so I can take only a chain. So I can delete remaining chains. Somebody asked yesterday how to delete, so I can delete the remaining chains. So I have to make it smaller actually. Okay, and then we have the tools. This binding, and it's called a dock trip. So what it will do is it will remove solvents, and if there are any ions, it will remove that. Then it will add hydrogen, it will add charges. All these things it will do. Generally, you can use it for docking, whatever be the software. Okay. First thing is I deleted, as in the quaternary structure of a protein contains many monomers, right? So here it's having say four monomers. COX2, it's a very big size. So I removed all of them. I just kept chain A, correct? As you saw. So you can do all these actually. That's the preparation of the document. Anyway, you have to do, it takes time. Yeah, now it's minimizing the energy. This will take time. Then you store that structure and use it for your document. That's very important. So directly from PDB, the PDBs will have many monomers. Okay, like if it is a 2 or 3 or 4 monomers connected, it will be difficult for docking because there are many docking programs accept only about 15,000 atoms. So you delete all of the various chains, number one. Number two, you add hydrogen, you remove solvents, and you add electrostatic, you put charges, all those things you have to do. That's what documentation. Yeah, that depends on some knowledge about it. Yeah, most of them will have. You must know where the active site is.
A lot of noise breaking.
So the problem comes only if the active site is between two monomers. Otherwise, you may have active site details in which monomer. It may be in chain A or chain B. So you keep that and delete rest. Okay. So this will take some time because it calculates the charges. So this will take some time. It's calculating the charges. There is a software called Amber, which calculates the charges here. So it will take some time. Okay, I did that before. So in Swiss model, can you see this Swiss model? I mean Swiss doc. You can see the Swiss doc. Can you see the Swiss doc?
Sir,
So in the Swiss doc, here I give the target, here I give the ligand, here I give my email, here I give some job name. Okay, okay, let's go one by one. So 1pxx is the 1pxx, and then aspirin I'm going to give, right? I have done everything. Yeah, it takes time. So I upload a file. So Swiss doc will check whether my files are okay. Yeah, this is very important. So after all the stuff which I have done using UCSF Chimera, I saved it. Then the ligand, I can give say as it says zinc or name of the compound. I can give it is there. Yeah, so it's got aspirin. So we can say okay. So both are okay. Can you see this? The PDB 1pxx is a cyclooxygenase 2 and aspirin, and then I give some name aspirin cox2 1pxx, and then I give my email ID. Were you all able to see this?
Clearly visible.
Yeah, then I say start doc, then my result will come in my email ID, just like your yeah. If it is an unknown ligand, you draw it in your Marvin sketch, get the three-dimensional conformation, store it as mol2 file, and then upload it just like I uploaded the protein. I can upload it, upload file. Okay. Okay. So new ligand, that's what I will do. Marvin sketch, you draw, minimize the structure, store it in your computer, and then you say upload file, just like upload target I did, right? So similarly here also upload file, and then I give that mol2 file. Please remember it has to be mol2 file. So that molecule will come there. Then you give your job name. Okay, so my results will come in my email ID. Okay, so like that. It will take some time, long time. Be available soon here. Okay, but actually it's still not available because it'll take a few hours. But I have run it yesterday. I've run it yesterday just to be on the safe side because it will take a few hours. Okay, so it's come like yesterday. Maybe you people can try it out. So this is the result I got. This doc. Can you please try it out? So it is giving. Can you see this?
We can see, yes, sir.
Can you click on the link I sent?
Yeah.
Is the link working?
Yes, the link is working.
Very good. So all of you please see. Look at the topmost one. It is giving cluster 0, 0, 1. It's giving you the energy minus 1.37. Okay. Oh, sorry. Topmost one, cluster 0. Delta G is given minus 1.52. You can rotate. Can you rotate the molecule?
Yes, sir. We can rotate the molecule.
Can you see the aspirin? Here, either acetyl salicylic acid. It will be in yeah here. Can you see here in my picture? So you also must be able to see.
So it's not visible in our link where we have where you have share.
Can you rotate the molecule?
Yes, so we can rotate the molecule.
Then you rotate and see, it will be there somewhere. If you can rotate, no, first you mark one button, top button.
Okay, sir. Okay, sir.
Then you will be able to see. Please mark one of those buttons. It gives you, as you can see, on the
Yes, sir.
minus 1.5, so it gives you the best delta G, because more negative it is, the best binding it is, right? So mark it first, and then you can see, rotate it. Now, if you mark it at different places, you will see aspirin in different places. Actually, try to mark different clusters. Okay, aspirin will be kept in different places. You can see that, right? You can see in different places. These are cluster 0. Put to cluster 1. You can see. Please try each one of the cluster and see where aspirin. So what the software does is it puts it aspirin in different groups of places, different grooves, and calculates the energy. Can you all see? Please play around with that. That will be giving you some now. See, now it says, in my case, aspirin has gone to some other place. That is in group cluster 1. So energy goes down, right? So minus 6.43, starting from high energy. So high energy means more like this.
One of the participant is sir asking with difference between the docking score, docking affinity, and docking energy.
Each software numbers will be different. Because I showed you first, if you remember, there are many non-bonded interaction terms, right? That is how energy is calculated. Electrostatic, hydrogen bond, van der Waals forces, dipole, ionic. So some softwares will assume dipole. Some softwares will not take ionic. Some softwares will take ion-induced dipole. So in that, see, so many things are there. So each software numbers will be different. So always use the same software. Don't do some compounds in one software and the set. It's totally wrong. So if you have one software, do everything in that software.
So she's asking docking score and docking affinity is the same thing or the different thing?
Docking energy is delta G, kilocals. Affinity is based on K, binding constant. Okay, so it's all derived numbers. Understand?
Okay, sir.
Delta G is equal to minus RT log K, right? So
And sir, docking score?
docking score is they come up with some number based on all these parameters. That is, each software will have their own numbers.
Okay, sir. Okay, sir. I think it will be clear to now. It's clear to
Use
Bhavna.
only same software actually. Because if some software gives score, try to use the same software. They will not give binding energy or they will not give docking energy. So always try to use. So score will be positive numbers. Whereas if you go for delta G, you will have more negative number. Okay, score is positive, higher the number. Understand? So try to use the same software for all your applications. Okay. I also did 2 more problems. Tripinavir. It's an antiviral drug. And as you know, proteases are enzymes which are involved in the replication process in SARS. Okay, so I took this particular protein 1P9U. It's a coronavirus main protease, 3LC protease, that is cysteine-like protease. It's involved in the replication process actually. So I used that 1P9A, sorry, 1P9U, and tripinavir is an antiviral drug. This was also tested in SARS-CoV the past 1 year. You must have heard about its name actually. Many drugs of antiviral, old antivirals were tested. Tripinavir, you can see selected non-peptide, it's used for HIV also. Okay, so I did that, tripinavir and 1P9U. Okay. Okay, I sent you the link. This is tripinavir with one of the SARS-CoV-2 protease. It's a big molecule. You can see it here in my picture. Please, I sent you the link so you can also open it. Can you open it?
We can open it.
Yeah. Can you see the tripinavir?
Definitely, sir. It's visible, sir.
Yeah. So it's a big molecule. Aspirin, when you saw that very small, tripinavir is a much bigger. Energy, look at the best one, minus 9.29, cluster 0, very close. Then click on, just it goes to different place. You can see different locations. So the software will put the ligand in different locations and then try to see, you see, they are just in completely different locations. This is tripinavir. Are you able to get it in different locations?
Sir, sir, I have one question, sir. If anyone wants to know the docking score of this protein ligand docking, then how will we check that? What is the docking score of this?
Delta G. No, this is the binding energy which is given here.
Okay. For this, there is a no. For this software, there is a no docking score kind of thing.
Hey, that is only 1 or 2 softwares used. Generally, this is the basic parameter. Using this delta G, everything is calculated. So this is most important actually. So this delta G gives you the energy, the binding energy. More negative it is. Docking score is calculated based on delta G. They put some plus or minus and then they get some number. Okay. So this is much more. It's like calculating CGPA from your percentage mark, right? Percentage mark is more important.
Sir.
That is the basic. Then CGPA is calculated by putting number of credits, this, and then they come up with 4.2 out of 5 or something. Right? But this is the basic. The thermodynamics is the basic part, right? So that is calculated based on various energy terms. Electrostatic energy. I showed you equations. So the electrostatic, van der Waals, hydrogen bond. So when you use that, you will get some kilocals per mole. So you add up all those. That's what you get here. and then you calculate docking score or docking energy, docking whatever you want to, interaction energy. Some people use interaction energy, like that, you know. People give
Thank you,
different.
sir.
This is a trypinavir. It's a long molecule binding to a 3-CLP, 16-like protease, which is very important protein in the replication, which is present, which happens inside the human body actually. That's being looked at to overcome this problem. Another one, again, another protease. CMPB. This is also a this is also a protease. 7-MPB. This is also a 16-like protease. And here I looked at another drug called nilfinavir. You must have heard of all these drugs in the past 1 year. Many of these drugs were looked at for the treatment of this COVID-19. Nilfinavir, immunodeficiency virus, HIV infection. Basically, they also work on the protease inhibition. Actually. Many HIV drugs also were tested for coronavirus, COVID-19 actually. It's a protease inhibitor. So here we have a drug called nilfinavir, another protease inhibitor, minus 9.18. Okay, I've sent you the link again, and you can play around with this. Yeah, this is called nilfinavir. Look at the structure. You can see it. You can see it. You got it. You are able to open nilfinavir.
Yes, sir. Participants are able to.
Yeah. Okay. That's what I want to see. So this is also big drug. Actually, antiviral drugs are big. You know, many of these antivirals. If you look at nilfinavir, remdesivir, fapinavir, all of them are quite big actually in size. When you saw aspirin, like that, very small. So you have the first step, the homology modeling. That means you compare sequences and then take the best template, and then you build your model and check whether model is good, store it. Okay. Then you go to UCSF camera, and then you can add hydrogens, you can remove solvents, you can minimize the energy. All those things you have to cut down on the size also, because many softwares take only 15,000 atoms. So make it smaller and then store it again. That you use it for your docking. So in the Swiss dock, you give your protein, whatever you have selected, stored actually. Ligand, if the ligand is already available in zinc, you can give the zinc database. Automatically it will select. Like somebody asked, if the ligand is not available, you have designed your own ligand. So go to Marvin and then draw your ligand, get the three-dimensional confirmation, store it in Mol2 format, then use that ligand. Then you give your email ID and the results will come in the email ID. And then you can see the result. You can look at the binding energy, which is given in the last column. More negative it is, that means more stable it is, correct? You can view this structure in the UCSF Chembial. You can do a lot of things for us. UCSF is much better in many things. I think it's done the job. Let's say downloaded file, we can so the same file we can view it under VCSF also. Yeah, and you can see this is your molecule written away. This graphical interface is very good and it can do a lot of measurements and all that also. This is a good program. So you can see your protein. So we can do some structural analysis. Many analysis can be done with the UCSF Chimera. See the protein and the ligand interaction. Actually. Okay, this is how entire docking concept works actually. Target sequence, template, multiple sequence, and this is the software actually BLAST, which does the job. We use Swiss model because it combines everything together. Docking validation is looking at Ramachandran plot. I showed you RMSD, All those are called validation. How good is your model? Is your data, is your model file size falling within the approved? How much is the variation between the template and the modeled protein, which comes in that Swiss model, right? If you remember, that's the validation part actually. Okay, then you see the, and then you have your protein, and then you have to go to UCSF to do a lot of things also, and make it ready. And then you have library of ligands, zinc or whatever database you have, you can get docking score, binding energies, good, whichever software you use, it gives you some number. Try to use the same software. So the and there is one more program which is called Swiss target prediction. If given a structure, you can predict what would be the target. So if it's a very new molecule, experimentally, if somebody has determined the target, it's okay. But somebody has, it's a new molecule, so nobody has determined the target, right? So that says target prediction is that way very useful. So you are designed a new molecule. You want to know what are the possible target of that molecule. Does it have any side effects? It's called Swiss target prediction. So we give a structure, it will predict the target. Give you a lot of information. So we can draw the structure, of course, or we can paint our smiles. So you remember. Let's do some other track. So it needs the smiles. So we can put in the smiles for this target prediction. Predict targets. This is a Vioxx, cyclooxygenase 2 drug. So it will give you various possible targets for the molecule. Okay, these are prostaglandin synthase 1 and 2 in the inflammation. So if you go and bind to both these targets, of course, okay. They are oxidoreductase type of enzymes. It will also go and bind to ketoreductase enzyme, some cytochrome P450. These are xenobiotic enzymes found in liver, which degrades, but the percentage is low. These are the most possible targets, as expected. We know that, right? I also know that. So target details are given here: PTGS1, PTGS2. We click on it. It will give you all the details about that target. Gene classification. It's called gene card. So it will give you all the details about that target. Okay. Prostaglandin G synthase 2, synthase, cytochrome, cyclooxygenase. It gives you the enzyme class. So many things it's going to give you actually. The gene, it gives you a lot of information about the gene here. Okay. Genomic data. Okay, that's on the UniProt. It will give you the UniProt number of that particular target. Yeah. So we can look at the UniProt. There is something called Chembial Database. It's a very good, a very important database which gives you a lot of properties, okay, of that particular cyclooxygenase 1. What are the synonyms? Organisms, it's from Homo sapiens. Then it will give you drugs and clinical candidate. Activity chart. Yeah, it will give you associated bioactivities, IC50 values. IC50 values is inhibition at 50% inhibition. It also gives you also assays also, how to do experiments. Okay, that's the beauty of it. If I have this enzyme, what type of experiments I can do to measure activity of a ligand. Then binding and affinity, all reported binding affinities. So a lot of information it gives about each one of the target. Lot of information. So even I can know what type of experiments I can do if I have some inhibitors I want to screen against this target. So the experimental details are also can be included actually. And any drugs are there against this target. It will give you those also. So these are the molecules that have been tested against that particular target. You can see this down, 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 down. As approved in 1999, FDA approved, like that, you know. So a lot of stories about the target can be identified. All these are Homo sapien targets. So if you are designing a drug for a cancer, for example, or if you are designing for a viral, for example, if you want to know whether there are possible human targets, this is a good program to run on. It will identify the human targets. For example, if you remember the previous page, yeah. So here I have given Homo sapiens. So it will identify if there are any possible human targets. And then it will give all the details about the human targets, the gene involved, in either ligands that have been known for inhibiting that human target. So a lot of information it tries to give actually. And any experimental assays that are available to study the target. So it's very useful for us to look at side effects, human side effects. If you are designing for virus or if you are designing for a tuberculosis bacteria, what type of human targets it's going to have. So this is a very useful predictive tool. So it's target prediction, extremely useful tool actually. So like you have all these new drugs for SARS-CoV-2 towards spike protein, towards protease inhibitor, towards helicase, towards all these. Those drugs we can also check in this target prediction to see whether it's going to have any human targets also involved. So that's the main advantage of this particular software. Actually. Okay, so how much time I have, Dr. Melu?
Time.
How much time I have? Because you wanted to have some.
So we still have half an hour. If there's something to discuss, sir, we can discuss.
Yeah, if there is any question. Anybody has
Participants, if there is any
any
query,
doubts? You have?
doubts. So one doubt I am having, that like if we have a ligand, and if we have a like if this system like protein ligand docking can be done with the autodock also.
Autodoc Vena. That's new. Yeah, you can do that. You have to do anyway. Ligand preparation is there. UCSF. Autodoc Vena also has ligand preparation. You can do UCSF Chimera also for ligand preparation, and then you can talk. Yes, no problem. But try to use the same software. Whatever software you use, try to use. Don't mix up. Don't mix and match, because everything will be different. The answer, no, whether it's docking score or whether it's docking energy or whether it is binding energy or minimum energy, those numbers will be different from each software.
More query I have, like if we have synthesizing a list of compounds and it is a novel compounds, we go to the Marvin sketch and we develop a conformers with the lowest possible energy conformer. Then can we directly save into the mol2 file?
I don't know whether it can directly, but you can save it in SDF and use OpenBabel, convert to mol2, right? It's not a big deal.
Okay, we can go to the SDB and then we can open Babel and then we can do the
Yeah.
mol2 file. Okay, sir. So one of the participant is asking, is
It
target
is unknown molecules also, because that is the whole idea. Known molecules anyway. If it is a known drug, the pharma company would have done all the side effects, right? So you just go to, I showed you on the first day, drugs DB, drugs.com, drugs, all those software, all those databases are there, which will give you all the side effects and all that. This is for a new molecule. Hold for drug, every data must be provided by the pharma company, right? So your life is simple. If it's a new molecule, suppose you are synthesizing in your lab and it has shown some good TB activity, I want to know whether any human targets are there in that. So I can use this. Correct? For existing drugs, I'm talking about drugs. Every data is available. You go to FDA database, you go to drugs.com database, you go to drugsDB database, you go all the information about drugs, around 3,000 or 4,000 drugs will be there. Otherwise, they won't get the approval, FDA approval. So don't worry. Only if it's a new molecule you are designing.
Any more doubts? Any participant is having a doubt?
And there is, okay, are there any doubts? I can just touch upon some other concept which is called pharmacokinetics.
थोड़े से तो है अभी कच्चे और पानी डाल दो
It's called pharmacokinetics. What the body is doing to the drug. We will know when we give it to animals or human, drug gets biotransformed, drug gets degraded, drug gets eliminated. Okay, so I just showed you one graph. You remember first class. So that's called pharmacokinetics. I'll not spend much time on that. Okay, so there is PK, pharmacokinetics. PD is pharmacodynamics. Pharmacokinetics, what the body is doing to the drug. Body is doing a lot of things. Liver has cytochrome enzymes, then there are lipases, there are oxidative reductases, esterases, hydrolases. So then biotransformation takes place, then gets eliminated, your drug, right? That's pharmacokinetics. And then what the drug does to the body. Suppose I'm looking at a infection, anti-infective drug, antibiotics I'm taking. So the drug is acting on the body, right? That's called pharmacodynamics. So this PK/PD is very, very important because that is where the body and the drug we are showing. Whatever I do in the lab is like I use cell lines, I use bacterial strains and study, right? You are designing some molecules, you are studying with the cell lines, different types of cell lines, mouse cell line, or if you are using bacteria. But in real life, the body also comes into the picture. Body means very complex. So many enzymes are acting on it. Okay, so many biotransformations taking place. So many, and then you have the elimination, urine-based elimination routes. So pharmacokinetics comes into the place actually. That you will know only when you give it to animal. I give a drug orally to an rat and see how the drug concentration increases with time in the blood and how the drug concentration goes down in time in the blood. That's the pharmacokinetics because of so many things, right? So we need to understand that there are mathematical models which can be used to predict how the concentration of the drug in the blood changes with time. That's called pharmacokinetics, actually. Okay, why is it important? It's important because you have heard about resistant strains. Suddenly, once upon a time, penicillin worked, but nowadays penicillin will not work for infection. They have started going into rifampicin. They have started going into okay, very strong antibiotics, right? Okay, vancomycins, like that, because the strains have become resistant. Bacteria has become resistant. So same drugs which worked once upon a time don't work. Not only in bacterial, many places this resistance has developed. So I may need to understand the concept of pharmacodynamics. Many times drugs are given in combination. If somebody has blood pressure, they are given ACE inhibitor and beta blocker, two drugs. If somebody has a blockage in artery, they are given some blood thinning agents like uh, blood thinners, aspirins, maybe for blood pressure. So like that, 3-4 drugs are given. So how each one of them act, that becomes a problem. Another thing is, no, drugs change given to. It works very well in my lab, but it will depend on the race. Drugs which work, for example, if you take chloroquine, anti-malarial drug, we all, maybe some of you might have taken for malaria. At least I have taken for malaria, this chloroquine, but it's very toxic to European, same drug actually. And then between men and women, there are differences. Okay, some drugs. For example, even if you take alcohol, ethanol, women are less tolerant to ethanol than men because they don't have the reductase enzyme which converts ethanol to aldehyde and so on, like that. So, pediatric babies versus geriatric old people, people who have renal impairment, okay, their kidney problems. So same drug works differently. Drug interaction. Healthy versus sickly person. A healthy person, nothing will happen. Whereas a sick person, something will happen. Okay, so this pharmacodynamics also changes a lot between all these aspects actually. Okay. So the PKPD you come to know only when the drug is given to animals. In the lab, you don't come to know about it because you are using some cell lines, you are using some bacterial strains, you are using plates or test tubes and doing all the experiments, but that is not the real life actually. Okay, so these aspects of PK and PD come into picture at that stage. So we need to understand some of it actually. There are many ways of modeling how the concentration changes in the bloodstream with time and so on. Actually. Another thing is dose selection. So sometimes doctor says take every 6 hours. Sometimes doctor says take every 12 hours, right? So you may wonder why. So again, it depends on how the drug gets eliminated from our body. Same drug may get eliminated faster in my body. Maybe it will take longer time to eliminate in a European population. Okay, so these type of things is very common. So I may take the same drug every 4 hours, European may take every 6 hours or 7 hours or 8 hours. Okay, and then labeling. If you see labels, a lot of information is given on the labels. I don't think we all read that. We just listen to the doctor and take the medicine. But if you look at the labels, a lot of information about pharmacokinetics. Don't take after this. Don't take when you are taking this medicine. Pregnant women should not take this. All this is to relate to the health of the patient, the biotransformation aspects, drug-drug interactions, all those, because of all those actually. Okay, so the labeling also comes into based on the pharmacokinetics actually. Okay, so typically this is how it goes. I take oral drug, goes up, and into the blood, depending upon how I get absorbed, and then it goes down, 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 because of biotransformation, because of elimination, all those things. So doctor will say take another drug after few hours, again it goes up, 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 comes down, down, like that, like that, like that. Whereas pharmacodynamics, you need some concentration for the drug to act. If you know antibacterial, there is something called minimum inhibitory concentration. If the concentration is higher than MIC, minimum inhibitory concentration, drug will not act on the bacteria. Even if there is some drug, it will not act unless that concentration is above the MIC. Okay, so there might be some drug in the bloodstream going round and round, but it might not kill the bacteria because you need that MIC to kill the bacteria. Okay, so that pharmacodynamics gets complicated because of this pharmacokinetics. You have to. Okay, so unless that concentration of MIC is reached in the bloodstream, bacteria won't get killed. But you are continuously having elimination from the body because of so many biotransformations. So bacteria will get killed only for some time if the concentration is high. And when the concentration goes down below that MIC, bacteria won't get killed, but the drug will still be there inside the body circulating. Okay, so that's the complications of PK and PD. Okay, so there are many approaches by which we do mathematical modeling. Okay, if you look at it, typically this is how it is, right? Goes down, reaches some maximum, C max you call it, then it goes down. Okay, there are many approaches. You consider the entire body as of the blood plasma as one compartment. So that's called one compartment model. The drug comes into the compartment and then drug gets eliminated. But then there is a two compartment model where you divide your body into two parts, blood part and maybe tissue part. Whatever drug is there in the blood only will get eliminated because the blood is getting circulated in your liver and liver does all the biotransformations. So whatever is there in the tissue will come to liver, come to blood and then only get eliminated. That's called a two compartment model. So we have one compartment models, two compartment models. So we use a lot of differential equations. I'll show you some of them. Yeah. Mathematical model is so if I am taking oral drug, drug gets absorbed and goes to, this is a one compartment model, drug gets absorbed and goes to the bloodstream. It gets eliminated because of biotransformation. So you put in some constant terms and then you represent concentration of the drug as a function of time using some differential equations. Okay, and then you can solve these differential equations also. You get some mathematical relations like this. So you have absorption orally, Ka, elimination, Ke. So you have two terms here, Ka and the Ke. And then the entire compartment is considered as V, volume. And then you are given a drug at some dosage, D. And F is your bioavailability, okay, our fraction that is getting absorbed. So we can, this is how you model this, and we can look at how the concentration will change in the blood as a function of time. So when I change Ka, that is absorption from my stomach into the main compartment, For different value of Ka, the graphs can go like this, like this. So if the absorption is very fast, the peak reaches very fast and you get very high peak values. The absorption is very slow, the peak reaches very slowly, and the maximum concentration also changes. So these are all happens only in the human body. Okay, this you cannot do it experimentally. So we try to model it actually. It's called a one compartment model, and based on studies with animals, we can get all these parameters and then we can develop this type of relationship actually. So by changing absorption constant, by changing elimination constant, I can see how this graph will go up and then come down. Okay, as you can see here. Actually. That's the beauty of this whole study of a one compartment based model. Then you can do even a two compartment model. That means another part is absorption, bioavailability. If the bioavailability is very high, maximum concentration is reached. The bioavailability is low, not very high concentration, very low concentration is reached actually. Like that, you can do a lot of simulations, they call it actually. So if you have two compartment model, let me know. You have say central compartment, only the blood, tissues like elimination is happening only in central compartment. There is some absorption, desorption taking place between central and peripheral compartments. Okay, then you can again mathematically model these. These are the differential equations. We can solve these. These are very simple differential equations to solve. So we can see how the concentration of the drug changes with time when I take a dose orally. Okay. So this is the entire concept of pharmacokinetics and pharmacodynamics actually. Which is generally useful when we talk about drug orally taken by a human. Then you can even go for, these are some solutions. You can go for even, yeah, two compartment oral absorptions. So different types of combinations, intravenous, two compartment, intravenous, one compartment. When you talk about intravenous, the whole drug is given directly into the blood. So there is no question of GI absorption also coming into picture. So all those various combinations can be mathematically modeled actually. Okay, anything you want to ask on pharmacokinetics and PD? I'm not going too much in detail about these because yeah, these are three compartment models and so on. Actually, you can make it very complicated, the whole human body. Look at how the drug gets eliminated as a function of time.
Any doubt? Any queries
Yeah, I'm not
from
going
the
too much
participant
into
side?
this, but it can be done because this is the realistic picture of how the drug and the body are interacting with each other. Whatever you do in the lab is only in vitro on a Petri plate or on a test tube, right? Or in a cell culture lab, but that is not the real.
I think it's clear. It's very much clear, sir. This pharmacokinetic model, and although we need time to understand this equations, and it's very new to us, so it will take time to understand these peripheral, central, and models.
That's why
But
I'm not
yeah,
going too deep into that because as a chemist, you would not be using much of this actually. So I did not go into that. I'm just superficially touching it because you have to know that real life is much more complicated than test tubes and Petri plates and computers. So, but this can be modeled nicely with a set of differential equations and so on. Actually.
We definitely try it out, sir, these using these softwares, and will definitely wants your guidance still after this workshop.
Yeah, sure.
Yes, participants, any doubt regarding all the 4-day session? So can we wind it up now?
Yeah.
No,
I
ma'am.
think
I think
no.
it's no. They have cleared their doubts. Thank you, sir.
So in a short while, we can start a valedictory session now.
Okay. Let's wait for 5 minutes, or
Yeah, sure.
yes, sir.
Just a 5 minutes break.
Yeah, 5 minutes break, and then we can start with the valedictory session.
Sure, sure, sure.
Thank you, sir.
Good evening, sir.
Good evening, Minu.
Good evening, sir. Sir, you can stop sharing your screen now. We will start with the valedictory session.
We can start with the valedictory session.
Excuse me, sir.
Mukesh, sir.
Mukesh, sir.
On 5 minutes break.
Wait for 2 minutes. Okay. shall we start with the validatory session? Yes, Nisha, you can start.
Okay, sir. Thank you, ma'am. Thank you. Thank you, sir, for your really valuable inputs and valuable information. And it's really a first time experience. Like we are really working on hands on these software, and we have experienced this software first time ever on online these kind of workshop, and we have learned so much of about the QSR pharmacokinetics and the drug designing. And today is the most it's a dynamic part, which is a molecular docking. So it's really gives a motivation for the researchers to start with a computer aided drug designing, and to deal with these softwares. I think it will be much more easier now to deal with these softwares, and we are not be like inhibiting these using these softwares. So I would like to invite Dr. Meenu Agrawal for giving the summary of the workshop.
Thank you, Nisha. Honorable Professor Mukesh Doble, worthy patron and principal, Dr. Krishan Kant, coordinator, Dr. Poonam Anand in absentia, members of the organizing team, Dr. Neha Batra, Mr. Chandar Atri, and Dr. Nisha Agarwal, all my colleagues, our most valued participants from various colleges, universities, research institutes across the nation. A very good evening to all. On behalf of Department of Chemistry, Agarwal College, Ballabgarh, and as a convener of this workshop, I am very pleased to be here to summarize this important event, National Lab Virtual Workshop on Computer-Aided Drug Design, conducted from 6th October to 9th October 2021, which was organized under the chief patronship of Shri Devendra Gupta ji and patron and principal Dr. Krishan Kant Gupta. The objective of this workshop was to make aware of how to design drugs with the help of computers. In the inaugural address, Dr. Krishan Kant Gupta encouraged the participants for workshop and emphasized on the need of such workshop in today's scenario. The technical session of first day was based on drug properties and databases, in which the resource person, Professor Mukesh Doble, introduced some databases which contain structures of large number of molecules, and large number of databases were introduced like Drug Bank, ChemSpider, ChemDB, PubChem, ChemMB, SwissAdme, and SwissAdme is a software where we can calculate theoretically some of the properties. Next, the concept of SMILES was introduced. That is the simplified molecular input line entry system. So in single line, we can describe the entire molecule. The technical session of second day was based on pharmacophore modeling, where we had the opportunity to learn the use of a software called Pharmagist, Zinc15, and OpenBabel softwares. These are the freely available web software for pharmacophore detection. It is ligand-based. Later on, we used PyMol, which is a molecular visualization system. Again, this is a free software and distributed by Schrödinger. Then after finding the common pharmacophore, we can go to a software called Zinc Farmer. We need to load features and can look at which compounds in the Zinc database had these type of pharmacophores. The technical session of third day was based on QSAR, that is quantitative structure activity relationship. That means to develop a mathematical relationship between the activity IC50, LD50 with the structural properties, structural descriptor, structural features used in QSAR. There are many softwares like ChemDes from which we can get thousands of descriptors. So this is an integrated web-based platform for molecular descriptor and fingerprint computation. Finally, we make us aware about the excellent features of Excel. The technical session of the fourth day was based on docking, where we were enlightened with the use of the softwares like SwissDoc and UCSF Chimera. So by the use of these softwares, we can get information about the docking, that is how the two or more molecular structure, that is the drug and protein, they fit together. So the protein interacts with the small molecules. So this is a very important part. In this 4 days workshop, about 45 participants from 15 universities, 23 colleges, and from 10 different states, they participated. And this data speaks itself about the success of this workshop. And I would like to congratulate and thank worthy principal sir, Dr. Krishan Gupta, patron of this workshop, for his inspiring guidance. He is the backbone of the successful workshop. I'm sure that everybody who has participated in this workshop is illuminated and full of new ideas. The deliberations and the interactions during the workshop will certainly help in augmenting the research activities and give a new direction to the young researchers. So thank you all for this 4-day productive workshop. Thank you very much and best wishes from Agarwal College, Balukar. Thank you.
Thank you, Dr. Binu. Now I invite our Honorable Patron and Principal, Dr. Krishan Kant, for addressing the valedictory session.
Thank you, Nisha. Good evening, all.
Good evening, sir.
Good evening, especially to our speaker, Sri
evening, sir.
Mukesh Doble ji.
Good evening.
As you all know, he is a very senior scientist, biotechnologist, and a versatile genius, I can say, from IIT Madras. Dr. Minu was talking about the success of the conference or the workshop. 15 universities. It is very encouraging. Then 10 states and more than 40 participants. Initially, when this topic was being decided, somebody said that this topic is very specific and we may not get the required number of participants. So this is the persuasion of Dr. Minu Agrawal and his team and attraction of the speaker that we could rope in more than 40 participants in this 4 days workshop.
Thank you, sir.
And that too, the timings, which are the leisure timings for the teachers in particular. Because after 2:30 or 3, they are usually at homes, busy with their kids, busy with their family or taking rest. But the credit goes to the speaker, Shri Mukesh Doble ji, that he could engage the participants in such a nice way. And as Minu was talking about, so I can guess this workshop was very educating, empowering, and enlightening. And all the participants who joined this workshop must have been benefited a lot. Chemistry, you have learned virtually. This is a wonder. Computer aided drug design. डॉक्टर साहब ने सब कुछ आपको सिखा दिया दूर से. So these are the wonders of technology. So technology is playing a very vital role. But you always say, and we always say, that technology is a boon as well as a bane. ये जो कोरोना 19 वायरस जिससे पूरा विश्व त्रस्त है, ये भी टेक्नोलॉजी का ही कमाल है. ऐसा मानना है लोगों का कि चाइना की वुहान लैब से जब लोग साइंटिस्ट कुछ कर रहे होंगे टेक्नोलॉजिकली तब ये लीक हुआ वहां से, ऐसा लोगों का मानना है. So today is a valedictory function. Everybody knows that every beginning has an end, but I am sure that this is a very pleasing end. और एक चीज और मैं आपको शेयर करना चाहता हूं कि किसी भी अच्छी शुरुआत के लिए अंत बहुत जरूरी है. Don't take it as a valedictory. It is not the end. Professor Doble will not be there with us on daily basis. Maybe when we need his expert advice, he may be available. So now you have to take the lead. All the participants who have joined this workshop from various universities and colleges, you have to take this concept forward in your own institutions. Please don't leave it here. Don't forget it. Whatever takeaways have been during these 4 days, during the very informative, illustrious, enlightening deliberations by Professor Doble, please keep all this information with you. Take it to your institution, take it to your labs. Start doing small things. Ask your principal, ask your management. Discuss with them. persuade them and have some funds for your labs and start doing research. Definitely, if you have the convincing power, then managements are always ready to support you. So this 4 days exercise, this 4 days words of wisdom, which have been showered over all the participants by Professor Dr. Mukesh Doble ji, they should not go waste. Please, please, my request is that keep it with you. आप इसको think over it. Inculcate a culture of research, maybe at a very small level in your institutions. And as I can foresee that since this workshop has been on virtual platform, you are well aware of all kind of softwares, which sir must have told you all, then maybe it may not cost much to start. And I always say that whenever you want to start a research work, please write a review paper. Because review paper in itself costs nothing, only your time and energy and your wisdom. When you write a review paper, you get the literature review and literature survey, starting from the very beginning to the latest, and you have an overall aspect and aerial view of the entire topic, and then you can start the work in your labs. So Minu, you asked me to be present during the valedictory session because I am an odd man out. I am a student of physics. I don't know chemistry. I don't know what is drug and what is design. और ड्रग शब्द वैसे भी आपको मालूम है बहुत खतरनाक है, और अभी देश में पूरे में हल्ला गुल्ला मचा हुआ है ड्रग्स को लेकर के, और नारकोटिक्स का जो डिपार्टमेंट है वो बड़ा एक्टिव है देश में. लेकिन मैं एक चीज और चाहूंगा सभी पार्टिसिपेंट से कि प्लीज डोंट कीप इट टू डिजाइन ओनली. Research is of no use unless it goes from labs to fields. And knowledge should go out from the books to the students. So please take this concept to fields. Research should be humanity oriented. It should benefit the humanity, it should benefit the humans. So please don't restrict yourself to the drug design. Please focus on delivery also. So it should be design and delivery. Only then this cycle will complete. I feel like this. So once again, on behalf of Agrawal College, Ballavgarh, on my own behalf, on behalf of our entire Department of Chemistry, including Dr. Poonam Anand, Dr. Meenu Agrawal, Dr. Nisha Agrawal, Neha Batra, and Chandaratri, and other colleagues, I thank our speaker, क्योंकि इसकी जो सूत्रधार है वर्कशॉप की वो तो मीनू है, लेकिन इसके जो संचालक हैं वो मुकेश जी हैं. तो मुकेश जी का बहुत हृदय से मैं आभार व्यक्त करता हूं, धन्यवाद करता हूं कि इतने एक हाई लेवल के इंस्टीट्यूट का एक एस्टीम्ड प्रोफेसर अग्रवाल कॉलेज के प्लेटफॉर्म पे आकर के, वो भी 4 दिन के लिए, लोगों को एनलाइटन करे, लोगों को इल्यूमिनेट करे, लोगों को एम्पावर करे. ये अपने आप में बहुत बड़ी बात है. तो धन्यवाद के लिए शब्द कम हैं, लेकिन फिर भी एक परिपाठी है, एक व्यवस्था है जिसके तहत मैं सर आपका हृदय की गहराइयों से स्वागत करता हूं और आभार व्यक्त करता हूं कि आपने मेरे शिक्षक साथियों को, भिन्न भिन्न विश्वविद्यालय से जुड़े हुए शिक्षक साथियों को इस संबंधित विषय पर उनका ज्ञानवर्धन किया है. और मैं आशा करता हूं कि भविष्य में भी जब कभी हम आपको याद करेंगे अपनी नॉलेज एनहांसमेंट्स के लिए, तो आप हमारे साथ वर्चुअल प्लेटफॉर्म पर, और यदि ठीक ठाक जैसा कि लोगों की मान्यता है कि अब 1-2 महीने में कोरोना चला जाएगा, और जिसको लोग आजकल कह रहे हैं न्यू नॉर्मलसी, अब ये न्यू वर्ड पता नहीं क्यों लगा दिया लोगों ने. नॉर्मलसी इज नॉर्मलसी, and you never know when this normal becomes abnormal. This we saw last year, and this year also in February and March. But whenever the situation will be normal, I would request you to visit our institute sometime so that we all can be benefited by your mere presence in our campus. So with this, I thank you, sir, and I also thank Dr. Meenu Agrawal. He could persuade me, because just now I came from college, but because it was her persuasion that, sir, you have to be there. So I am here with you all. And I also thank all the participants who are on board with us from various universities and colleges. क्योंकि मैं ये कहता हूं कि वक्ता चाहे कितना भी उच्च श्रेणी का हो, लेकिन यदि श्रोता नहीं है तो वक्ता की कोई कीमत नहीं होती. तो the value of the speaker is decided and judged by the audience. So we had very good quality audience. So I also thank all those participants also. By their participation only, we could make this workshop as a success. And I also thank and congratulate the faculty members of my chemistry department that they could organize a workshop of such a high dimensions. And I wish that in future also they would continue to do so. Thank you very much. Thank you. Jai Hind. Over to Meenu, please.
Thank you, sir.
Thank you, sir, for such an illuminating and guiding guiding views for which will really like force for the researchers. Thank you, sir. Like
Nisha, can we have now feedbacks from the
Yes, yes.
participants?
Yes, definitely. So many of the participants wants to speak. Like, can we unmute all?
Yes, sir. Participants, now you can unmute yourself.
Ma'am, good evening, ma'am. Good evening, ma'am.
Good
Hema
evening.
Prabha from Borderland University.
Okay.
Okay.
So
This is
she's from Assam.
Yes, ma'am. Yes, ma'am. This session was very nice. I like it most. And Sir Mukesh has also spoken so nicely that we all can understand very smoothly, and it is very valuable for us. Thank you, ma'am. And thank you, Principal Sir of Vallabhgarh Agrawal University College. Thank you, ma'am. And also thanks to all the organization or all the organizers of this national level workshop that
Thank you.
all of you organized very nicely. Thank you, ma'am.
Thank you.
Dr. Binu, can I say something?
Yes, ma'am. Yes.
Yeah, I'm Dr. Abhilasha Shauri. I'm from Manav Rachna, Faridabad. So, ma'am and Sir Gupta, sir, thanks for allowing us to participate in the workshop firstly. And I just don't want to miss the opportunity to thank Mukesh, sir. And Dubli, sir, just wanted to tell you I'm a big fan of yours. I have literally no knowledge in the field, sir. But 2 years back, I tried to join your program on NAPTEL. And since then I got to know, I really got interested into this field. And sir, the comprehensive view that you have given, that actually has opened up my mind at least. And I was really afraid of going further into this domain. But since I'm from plant sciences, sir, I would definitely like to find out more on the suitability of phytochemicals as the lead compounds. And you have really shown us the way, sir. Thank you from the bottom of my heart. Thank you, sir.
Thank you, ma'am. So this is on behalf of Principal Agarwal College Vallabhgarh, on behalf of all organizing team and all the participants. Please accept this token of gratitude.
Meenu, again virtually.
Yes, sir. So please accept this virtual certificate of gratitude.
Thanks for this.
Participant, please mute yourself.
like I would like to invite Dr. Neha for giving the vote of thanks.
Thank you, Nisha ma'am. Yeah. Good evening, everyone. I, Dr. Neha Batra, deem it a great honor and privilege to propose the vote of thanks on this occasion. A heartful vote of thanks to our speaker, Professor Mukesh Doble, who spared his valuable time from his busiest schedule to grace this workshop with his immense knowledge and expertise. So, this workshop has been one of its own kind, and I am sure that each one of us has gained something or the other. We are grateful to our principal and patron, Dr. Krishnakant Gupta ji, for his words of encouragement. His able guidance has always encouraged us. I thank him for his wisdom, vision, and indefinite support. My heartfelt thanks to the coordinator, Dr. Poonam Anand, and convener, Dr. Meenu Agarwal, for organizing this virtual workshop. A special thanks to Dr. Meenu Agarwal, who planned and organized this workshop from a scratch. I owe my gratitude to all my colleagues who have worked hard to ensure that this workshop is a success. Last but not the least, a special thanks to all the participants for showing their keen interest and making this workshop a success. Thank you all.
Thank you, Dr. Neha. Thank you, and all the thanks to all the organizing team. Now we will end this workshop with the Kalyan Mantra.
सर्वे भवन्तु सुखिनः सर्वे भवन्तु सुखिनः सर्वे सन्तु निरामया सर्वे सन्तु निरामया सर्वे भद्राणि पश्यन्तु सर्वे भद्राणि पश्यन्तु मा कश्चित् दुःखभाग् भवेत् मा कश्चित् दुःखभाग् भवेत्।
Thank you, sir. Thank you, everyone. Thank you, everyone, for the from the bottom of our heart. Thank you
We have
very
shared
much. This feedback link has been shared to you, and now we will share with you link for test. So there are total 15 multiple choice questions. So you have to attempt in 10 to 15. We will give you 10 to 15 minutes for attempting this test. Nisha, you have shared this link for
yes,
test?
Yes, ma'am. Yes, I have just shared the link.
This is link for test or feedback form?
Ma'am, this is first I have shared the feedback form and then I have just sent it the link for the test.
Okay. So
I think this is the link for the test. Yes, ma'am, it's a test.
Okay. Thank you, sir. Thank you.
Thank you very much.
Thank you, sir. Thank you so much. Thank you.
मेरी छूट अब परिवार तुम्हारा कन्यादान